All right. While we work on getting our other presenter through some technical issues, I'm going to go ahead and get started. So, like I said, welcome back, everybody. This is Brandon Houle from MHRS, Viral Hepatitis Unit. It's my pleasure today, or it's been my pleasure to put together this session on drug analysis. So I'm going to go ahead and give you a brief introduction of our first speaker. So Elaine Doherty is a forensic scientist manager at the Michigan State Police Bridgeport Forensic Laboratory in the Controlled Substances Unit. Elaine has a bachelor's degree in biochemistry from Michigan Technological University and completed coursework in Michigan State University's Master of Forensic Chemistry program. She's worked at the Bridgeport Lab for 18 years and has supervised the unit for 7 years. So please give a warm welcome to Elaine Doherty.
Thank you, Brandon. I'm going to try to share my screen here. Hang on. A little bit of finagling here. I have 3 screens going.
Understandable.
I will figure this out. There we go. All right, are we good?
Looks good.
All right. So as Brandon said, my name is Elaine Doherty. I've worked at the Bridgeport Forensic Laboratory for over 18 years now. I work in the Controlled Substances Unit, and what we do here is we get in samples from police agencies that they suspect are drugs, and then we use chemical analysis to determine whether or not a controlled substance is present. The job is really interesting. Things change pretty quickly in the drug world, especially in the past 10 years or so. And so it's kind of fun to try to keep up with things, and it's kind of fun to try to look for new trends and that sort of thing. So I've been delving into that in the past few years. And I just want to share with you guys some of the trends that we've been seeing so that you have an idea of what's out there right now. And then also just some additional information about some of these different drugs. So first of all, I don't have any financial conflicts here with anything that I'm going to present. And the Michigan State Police have 7 laboratories within the state of Michigan. We have 2 in the Metro Detroit area. We have 1 in Lansing, 1 in Grand Rapids. We're there in Bridgeport. Grayling covers most of the northern part of the Lower Peninsula. And then Marquette covers most of the Upper Peninsula. And Michigan's a pretty interesting area for drug trafficking because it's almost a dead end. So besides the large amounts of drugs that are crossing the border into Canada, we're not really a through point to anywhere except those large amounts going into Canada. So it is really interesting. Almost all of our larger cases come from the Metro Detroit area. Very few come from any other area in Michigan. I put these initials of the labs up here. You might see these later on. I'll try to refer to them by the name of the lab when I reference them. So like I mentioned, Michigan is pretty interesting for drug trafficking. This is a graph that actually shows the types of drugs and the percent of the total items analyzed for each of those types of drugs. And as you can see there, out of our 7 labs, 4 of our labs predominantly see methamphetamine coming in. And you can see 2 of those labs, actually over 50% of their submissions contain methamphetamine. You can see our Metro Detroit lab sees a significant amount of cocaine. And then opioids are pretty standard across the state. There's a little bit of variation there, but opioids are pretty consistent across the state. So you can see just by this simple graph that Grayling, Grand Rapids, Lansing, and Marquette, those areas are predominated by methamphetamine. Metro Detroit and Northville see more cocaine. And then Bridgeport, we seem to be somewhere in the middle where we get a lot of everything. And it's just in the past probably year or year and a half where we're seeing more methamphetamine than cocaine. As an example, in 2015, only 3% of our identifications were meth, and now we're up to 30%. So that's a really rapid change for us as far as methamphetamine goes. So statewide, methamphetamine is 36% of our total items analyzed. It's always been a bigger drug on the west side of the state, but it is starting to move in, well, it has moved in to the east side of the state, except for the Detroit area. As you saw in that last graph, the Detroit area still really isn't getting a whole lot of methamphetamine. But with the meth trafficking, I would not be surprised if that changes soon. One big change for meth for us as well is that most methamphetamine samples we see are pure. So they're almost always crystal meth. They're almost always 100% pure and hardly ever have anything else in them. As far as cocaine goes, this is an interesting one. About 19% of the total items analyzed were cocaine. By the way, these statistics were from the beginning of, from the first 3 months of this year. So these are very, very current statistics, and the way drugs are changing so rapidly, I think that's really important to have what's actually out there more or less right now to share with people. Something else I meant to mention and forgot were some kind of qualifications about this data. So this is data of samples that were analyzed, and their samples, we don't always analyze everything that comes into the lab. So for example, if we get 4 white powder samples coming into the lab, we may only analyze 1 of them. This also depends on what the agencies submit. A lot of agencies don't submit everything, or maybe they wait to submit it until it's going to be going to court. And then the other thing is some of these things are submitted quite a length of time after they were actually seized. So if we're looking at data from January, February, and March, those were likely seized in December, January, and February, but there's probably some in there that were seized 6 months, even a year prior to that. So as much as we'd like to be real time as far as what's out there, there is a little bit of a lag in the information. So 19% of what we analyzed contained cocaine. Now, one big change we've seen, and I've talked to quite a few people about this, and no one really knows what's going on. We're seeing a lot more pure cocaine samples, and that's exactly the opposite of most drug trends. Usually, kind of especially at the end of the drug dealing or the drug trafficking routes, usually cocaine samples were 40 to 60% pure, but in the past 6 months or so, I'd say about 80% of our samples are 100% pure. And this includes even small amounts, 1/10 of a gram of cocaine. Used to virtually never be pure, and now it's a regular occurrence for that cocaine to be pure. Now, opioids are, of course, of a lot of interest, especially with the overdose risk associated with them. 16% of the total items analyzed by our lab contained at least 1 opioid. And you can see in that table there, the majority, 67% of those samples contained fentanyl. Only 3% contained heroin with no fentanyl. 26% contained a combination of the two. And then that 4% there, those are actually a fentanyl analog called fluorofentanyl. You may have heard of parafluorofentanyl. Fluorofentanyl is kind of a general term for that I'll be using. We don't specifically identify if it's parafluorofentanyl, but it likely is. So what this means is 93% of the opioid samples analyzed contained fentanyl, and 97% contained either fentanyl or a fentanyl analog. Again, this has quickly changed over the years. I mean, we used to have a small percent of samples with fentanyl, and it just keeps increasing. My kind of theory on this, and I have no idea if I'm right, just to admit that. My theory on this is that heroin is still made from a plant. So they grow opium poppies and eventually it gets converted into heroin and then it's sold. Where fentanyl is simply made in a factory. You buy the chemicals, you mix them together, and you can produce kilos of fentanyl very, very easily. This is reflected in the price as well. A kilo of heroin can be as expensive as $80,000, where a kilo of fentanyl can be around $5,000. So I would not be surprised if the appearance of heroin continues to decrease. And to be honest, I wouldn't be surprised if eventually it disappears altogether. And there's the comment about the neither block there being fluorofentanyl. So another interesting thing is the distribution of opioids by lab. So here you can see the different labs and what was present in the samples from those labs. So the blue are the labs with heroin only samples, and you can see Marquette actually had more of those than any other lab. Most labs are in the single digits as far as the number of samples with just heroin. You can see that mixture of heroin and fentanyl is prevalent basically everywhere except Bridgeport and the Grayling lab. And those two labs seem to be the ones that have more samples with fentanyl only. And then the little yellow tab at the top is that fluorofentanyl, like I was mentioning. Because Marquette has relatively few opioid samples, I think that yellow, even though it looks like it's a large percent of samples, I think it was only 3 or 4 samples altogether. So 4 of our opioid samples also contained a benzodiazepine. These are a couple of years have been around. So these designer benzos are not approved for pharmaceuticals, but they're being made mostly in China. We see them alone, we see them in counterfeit tablets, and then occasionally we see them in opioid samples. When we see them in opioid samples, they seem to come in waves where we'll see kind of a cluster of them for a month or 2 and then it'll go away again. And it seems like we might be in one of those clusters where we're starting to see those again. Last month we had a few more samples pop up. Now the importance of this is that benzos, especially these designer benzos, so these are basically more potent forms of something like Xanax. They're very sedating, and Narcan does not work with them. So if you have this in a sample along with an opioid, it can definitely complicate the treatment of someone who's ingested that. Fluorofentanyl, as I was mentioning, is more or less the only fentanyl analog we're seeing right now. We're seeing it by itself sometimes, not very frequently, but we are seeing it by itself sometimes. But most of the time it is mixed with heroin and or fentanyl. So there's the numbers of samples there. These are actually fairly steady from the end of last year. The number of opioid samples or the number of fluorofentanyl samples with no heroin or fentanyl is up, though. It was only 14 for the fourth quarter of 2021. So we're seeing more fluorofentanyl by itself, but the opioid samples with fluorofentanyl has stayed fairly steady. One other fentanyl-related substance that was detected in samples, and that's acetylfentanyl. I've heard rumors there's a carfentanil sample out there somewhere, but either it's not been submitted to the labs yet or we haven't analyzed it yet. So here's a graph showing those trends of fluorofentanyl. The top orange line is the mixtures containing fluorofentanyl, and then the blue line at the bottom is fluorofentanyl with no other opioids present. Another group of opioids, or another compound or class of compounds we're seeing in opioid samples are these compounds called nitazines. And they all have fairly complicated names, but the common thing they have is they all end with nitazine. These are opioids. They're non-fentanyl related opioids, and they are really prevalent in some areas of the country. We see them occasionally, but not too often. And last quarter, we saw 2 different nitazine compounds, and they were both in counterfeit oxycodone tablets. And I believe for both of those, they were, oh no, one was an M30 and one was a different marking. I don't remember what it was. One of these was in the Metro Detroit area, and one was actually up in Grayling, which is pretty interesting. And since then, so last month and this month, we've had 2 more nitazene compounds pop up in counterfeit tablets. We had 1 fentanyl sample with 3-hydroxy-PCP. PCP is a hallucinogen. We had one of these last year as well. These kind of seem to be one-off samples, not sure how it happens, but we do occasionally see some kind of random substance mixed in with our opioids as well. And then tramadol is something that we continue to see in opioid samples. It may be in as many as 20 to 25% of our opioid samples. It's usually not very much tramadol, but it is kind of surprising how often it does show up. And then something that I've heard a lot of talk about lately is the compound xylazine. Xylazine is an anesthetic, so kind of like those designer benzos, it has sedating properties. And we're seeing that semi-regularly in opioid samples. It varies between 10 and 20% of the samples contain xylazine. Some are very, very small amounts, and some are more significant amounts. Looking at this graph though, so this graph starts in January 2021, and it goes through actually last week, and it shows the percent of opioid samples that contain xylazine. And you can see if you kind of drew a line through this graph, that it seems like the amount of xylazine in samples is increasing. So the middle of last year, it looked like it averaged around just under 10% of samples contain xylazine. And right now we're looking at probably an average of around 15%. So that is increasing. It's still nothing near where the East Coast is right now. The East Coast, they have xylazine in almost 100% of their opioid samples. And they're also seeing xylazine by itself, which we have never seen here in Michigan. So they're seeing xylazine without any opioids present. So one thing that I like to do is see if there's any kind of trends here. So I just picked 2 weeks this year, and I looked at the samples that contain xylazine to see if there was anything similar in all of them. For example, do they all have tramadol in them, or do they all have diphenhydramine in them or something like that? And what I found out, and what you can see here, is of the xylazine containing samples from 2 weeks, you can see that there's a whole wide range of mixtures that contain xylazine. The one thing that is fairly common that you'll see here is fentanyl, diphenhydramine, quinine, and xylazine. So those first 3, fentanyl, diphenhydramine, and quinine, that's our probably our most, quote, popular sample we see are those 3 things. So to see xylazine in that is not that unexpected. But you can see from some of these how complicated these samples are and how many different substances are present in them. So for example, the one from Macomb County toward the middle, we have heroin, fentanyl, and fluorofentanyl, so all opioids. We have acetaminophen or Tylenol, caffeine, diphenhydramine, which is also known as Benadryl. Then we have another opioid, tramadol, quinine, and then we have xylazine. So these samples are very complex, and they have a lot of different kind of moving parts within them. And I kind of keep an eye on this still, and that still seems to be the trend, that they're all kinds of different mixtures. There's no real obvious trend to any of it. Even the ones all seized in Wayne County, you can see that every single one of them is different. So some of the things that we see our opioid samples mixed with, they usually have what I kind of call it a base. Like the bulk of the sample is usually made up of either a sugar, a starch, polyethylene glycol, or Miralax. And I'm not sure why they use that. I think it might be cheap. Sometimes we'll see creatine. So the first 3 lines are usually kind of the bulk of an opioid sample. And then the following 5 things, diphenhydramine, quinine, lidocaine, acetaminophen, and caffeine, we see in as more like a cutting agent of some sort within the opioid sample. And by far, diphenhydramine is the most common. Quinine is the second most common. Now, we do see a whole variety of things in addition to those compounds. Dimethyl sulfone is one. Triacetyn and phenacetyn are interesting because they're not even sold in the United States. They're not FDA approved, but they are FDA approved in Europe, in a lot of Europe, and also in Mexico. So those are probably not originating in the US. And then sometimes we'll see benzocaine, a compound called metamizole. And then gabapentin is an interesting one. So this is a prescription medication given for neuropathy. And we're seeing that not completely rare. Several times a month, we're seeing that in opioid samples. And then the quetiapine is a relatively new thing, and it seems to be actually increasing in frequency as well. So this is an antidepressant drug. So moving on to a different category of drugs, we're going to talk specifically about those designer benzos I mentioned that are mixed into some opioid samples. The other place that we really see these are the counterfeit alprazolam tablets. So the Xanax bars, or Xanny bars as they kind of call them on the street, right now almost all of those are counterfeit, so they don't contain the alprazolam they're supposed to. But almost all of them contain one of these designer benzos. So the primary place we see these compounds is in those counterfeit pills. We occasionally see them in a liquid form or in unmarked tablets, but that's not very common, probably a couple times a year. And then this is a list of these designer benzos that we saw in the first quarter of this year. You can see they have fairly complicated names. They're fairly complicated molecules as well. And they seem to be pretty sedating, and so they will, these are associated quite frequently with traffic accidents, OUID cases, and that sort of thing. So diving a little bit more into the counterfeit tablets, in the first quarter of this year, there were 127 counterfeit tablet items that were analyzed at the labs. 71 of those, so the largest amount, were the counterfeit alprazolam. They usually contained one or more of those designer benzos. The interesting thing here, though, is we only saw 46 real alprazolam tablets and 71 fake ones. So alprazolam, they can be round tablets and they can be oval tablets, and then they have the rectangular bar-shaped tablets. So far, the oval and round ones have been real most of the time. We've seen a few counterfeits. So those ones are typically real, but it's those Xanax bars that are usually the fake ones. We also saw 18 counterfeit amphetamine items. Those almost all had methamphetamine in them. And I think here it's really a cost thing. Methamphetamine is very, very cheap right now. The pill presses are very cheap. The dyes to press the pills are very cheap. And so I think that's why methamphetamine in pills is happening. One of the counterfeit amphetamine pills did contain tramadol, though, and one contained a designer drug called 4-fluoromethylphenidate. So this is an analog of methylphenidate. And then the last category of counterfeits we saw last quarter were counterfeit oxycodone. These almost always have some sort of opioid in them. And you can see here 35 of the 37 had some sort of opioid. Most of them had fentanyl. A few were fluorofentanyl. And then this is where those new drugs, the nitazines, are popping up. Ecstasy tablets are pretty interesting. When I started this job, ecstasy tablets always contained MDMA. And if we look at the statistics so far from this year, you'll see that only 1 out of 62 actually contained MDMA. Most of the ecstasy tablets right now are methamphetamine. A small number contain a drug called Eudalone. Eudalone is a designer cathinone. And if you remember the bath salt drug days, this is an example of a bath salt drug. So we're seeing that occasionally in the ecstasy tablets. And then N-ethylpentalone is another bath salt type drug. But it does seem like in the past 18 months or so, this has shifted even more to methamphetamine. For a while, it was about 50% Eudalone, 50% meth, but now it's shifting even more toward meth. And again, I think cost is a huge factor. Now, one class of drugs we're very lucky in Michigan to not have very much of are the synthetic cannabinoids. So again, probably about 10 years ago, these started on the market as kind of categorized as synthetic marijuana. We had a lot of them at that time, but since then they've really kind of tapered off. These usually have very, very bad effects to them. A lot of people that have tried them really don't like the effects, and I think that's one of the reasons we don't see them very often. But again, the East Coast has a huge problem with these. So last quarter, we saw 16 samples containing a synthetic cannabinoid, but the interesting part was that all of them were seized in a correctional facility. And then these are the names of the two synthetic cannabinoids that were detected. And they're almost always on paper. And they're not sheets of paper typically. They're usually little tiny pieces of paper that are being smuggled into the prison. I believe all 16 of these were the same correctional facility, and they have, I believe, arrested 6 people and are investigating 3 more employees of that prison for delivering drugs. They still have a major problem, but they are trying to figure out what's going on to stop this. And then last, we have kind of these one-off compounds, things that we've seen once or maybe twice that are pretty unusual for us. First is that fluoromethylphenidate that was in a counterfeit pill. And then we have a couple more bath salt type drugs. So these are methamphetamine and ethamphetamine analogs. And then this is one I had never even heard of before, 5-methylmethiopropamine. All 3 of these here, the last 3 here were all powders that were intercepted in shipments from overseas. So these were not specifically being sold in Michigan. These were ordered on the dark web from someone, and they were purchased directly from China, likely. This pentobarbital case was pretty interesting. It was a suicide of a teenager, and he actually ordered it from a dark web suicide website. So the barbiturates used to be real common, but we almost never see them anymore. I had a pentobarbital suicide probably about 12 or 13 years ago. So a pretty uncommon thing to see, but we did have one in the first quarter of this year. So that's kind of a breakdown of what we see in our samples. I'm trying to get the information out to people that it would be useful to, especially if we start seeing some new emerging group of drugs. Nationwide and even internationally, there's another group of drugs that are becoming popular that we just really don't see. They're called tryptamines. And so I'm kind of keeping an eye out for those. Those are hallucinogens. As far as overdoses, I don't think that would be really a factor. They would be really a factor in overdoses. These are things that are similar to the drug that's in hallucinogenic mushrooms. So something like that. And that is all I had. I will close this out, and then I will answer any questions you guys have.
Thank you, Elaine. And yeah, we're just gonna use the last few minutes after Mary speaks and go back to the questions.
Perfect. Thank you.
Yeah, thank you. And yeah, my pleasure to introduce Mary Fagat. I really apologize if I pronounced that wrong.
It's close enough. It's Figget.
So I got a little bio for you here. Mary's an epidemiologist at UNC Chapel Hill. Her research focuses on understanding and improving health outcomes among people who use drugs through community partnerships. She works in close collaboration with the North Carolina Survivors Union on their drug checking program, as well as other community-driven research projects. Mary received her MPH in epidemiology from Drexel University and is currently completing her PhD in epidemiology from UNC Gillings School of Global Health. Prior to beginning her doctoral training, she worked as an epidemiologist in local and state government, where she led several community-based projects, including a partnership with syringe service programs to increase hepatitis testing and vaccination capacity. And without further ado, I'll turn it over to you, Mary.
Okay, great. All right. Okay, and can you see my main slides, or is it also the notes?
I just see the main slides.
Okay, great. Well, I'm just going to go ahead and get started. All right. So yeah, today I'm going to be talking about public health drug checking, what that kind of means, and what we're doing in North Carolina. So just to start, I have no financial or non-financial relationships to disclose. And so I work at, as Brandon said, I work at UNC. I'm a PhD candidate and also working on a few harm reduction related projects. So this is our team. We were based out of the Injury Prevention Research Center at UNC and Gilling School of Global Public Health. And we have a collaboration with the North Carolina Survivors Union, who runs a drug user health site in North Carolina. And they had, to kind of give a little background on our work, they had obtained a infrared spectrometer a little over 2 years ago. And it really started all of this work where we recognized how important it was to be able to get drugs tested and what that meant for people on the ground. And so we're also affiliated with the Opioid Data Lab. Feel free to check out our website. We have a little more information about our drug checking work and a few other projects. So just to start, we're going to think a little bit about what information means when it comes to consuming substances and how that translates into health-related outcomes. So this is a picture of a few different types of beer. And in this picture, you'll see that we have information ready at our fingertips. So for example, one of the beers is this kind of funky beer with a few different flavors infused into it. There's pineapple, there's sweet cherry, there's different types of sugars. And then in the two other beers, we can see some other information that's available to us, like the percentage of alcohol by volume. And so when we're deciding what to do, if we want to, decide to have a drink, we can make decisions about what, maybe how strong of a drink do we want in this moment? Do we have to drive somewhere? Or, there's a choice here where we can pick between a drink that has lower ABV versus one that has higher ABV. Maybe someone has an allergy to pineapple, so the one all the way on the left is not a good choice. So this information is something that we often take for granted, and it is readily accessible to us. Something else that is becoming more readily accessible is as states are legalizing and regulating cannabis-related products, we have information about the potency and active substances also at our fingertips. There's also other information about kind of the subjective effects of the different products. And so when we decide what type of substance we might want to take, we have this information to also inform our decisions. And then there's other sorts of drugs. In this case, we might know what type of product we obtained. However, there's so much information that we don't know. We don't know about the potency. We don't know about what cuts might be present within the drugs. And so this makes it really difficult for us to know what might be best for us in that moment. So that's where drug checking comes in. And here, these are 3 different types of technologies that are present for drug checking related purposes. So many of us are probably familiar with fentanyl test strips. The picture in the middle is a Fourier transform infrared spectrometer. This is what the North Carolina Survivors Union uses. And the picture all the way on the right is a gas chromatography mass spectrometer. And all 3 of these technologies have existed for quite some time in different settings. A lot of them have been developed and utilized in the very beginning for more punitive purposes. However, what we're doing with public health drug checking is to repurpose these technologies to put them in place within communities to serve the needs of people who will benefit the most from this information. So just to talk a little bit about each of these 3 technologies, the 3 of them really all work in combination with one another. There's not necessarily one that's better. It really depends on kind of a cost-benefit analysis, a very informal cost-benefit analysis. So fentanyl test strips, I'm sure a lot of people here are very familiar with these, as I mentioned, but they tell us about the presence or absence of fentanyl It's inexpensive, it's pretty easy to use, and there's quick turnaround for testing. So we can test a drug sample within, almost like a couple minutes and get the results back to tell us whether fentanyl is present. Now, there are some challenges with some false positives that might appear on fentanyl test strips, but generally speaking, they're beneficial in settings where for a drug sample that we might not expect fentanyl to be in, it provides us with helpful, quick, inexpensive information. The other technology that's gaining in more popularity and is becoming more prevalent in the United States is the Fourier transform infrared spectrometer. It's the little machine on the bottom. And this is kind of a next level where it gives us point of care sort of information. So these types of machines can be set up within certain services programs or other harm reduction organizations where people can bring in a drug sample. They only need a tiny little amount. You can put it on a little crystal on the machine, and you can get results of what is in your drug sample within 15 minutes or so. And it really provides us with this wide range of thousands of different types of substances, including fentanyl, but also some other ones that we're starting to see, like xylazine. So it does provide us this comprehensive range with a pretty quick turnaround, but there's some limitations to consider. It's kind of expensive to get started. It's about $40,000 in order to purchase the machine. But once it's obtained and the library, some of the data on the back end is purchased, then it has a little less cost moving forward. And it also does take training of the operator. However, people don't need a chemistry degree in order to operate this machine. You can get trained and be able to run the machine. And, if you just kind of follow someone else who has expertise or, has been running the machine for some time. And then the last technology that we've been using in North Carolina is a gas chromatography mass spectrometer. And this provides us a very, very detailed amount of information about what's in a drug sample. It can detect tiny, tiny, tiny little specks of certain substances within a sample. It can tell us about different opioids, their byproducts, different fillers. So it gives us this really wide range of substance detection, and is much more sensitive than the FTIR machine. But it's really expensive, it's not as timely, and they are pretty much kept within these formal labs. So it can't be placed within community settings as easily as some of these other technologies. But they can be used in combination with other ones. So say if someone brought in a sample and it looks like, let me back up. So if someone brought in a sample to be run on a point of care machine like the FTIR, but we think there might be some sort of substance that we can't identify within that sample, we can send it off to our lab at UNC, which will do gas chromatography and be able to pick up some other sort of substance that we couldn't identify before. So I'm going to talk a little bit about what it actually means to run one of these programs, specifically talking about some of the benefits, and then kind of how we've situated ourselves as we started to grow. So overall, the benefits of drug checking include a pretty wide range of things. Overall, drug checking tools expand our knowledge to reveal what's actually in the drug supply. Studies have consistently shown that drug checking encourages safer drug use practices. And through this information, people can address their drug use behaviors, make informed decisions about their health, and implement other harm reduction practices that work best for them. And so these are three of our principles at UNC that we've tried to keep in mind as we've expanded and make sure that this is kind of the core of our program foundation. And we've realized that this is important, and sometimes I'll talk a little bit more about what our program is actually doing, but we've realized that as we've started to expand, it's easy to kind of get carried away and off in different directions. So we need to oftentimes recenter ourselves to make sure that we're serving the community in the best way that we are able to. And so at the individual level, drug checking really needs to be a service. It needs to be a service to people who use drugs. It needs to be based in a setting where they can obtain the services at a way that works best for them. Noting that sometimes the services, we might not be able to get a point of care machine out to some of our more rural communities. So maybe a mail-in drug checking service might fit a little bit better for that purpose. And then at the community level, drug checking, when we start looking at all of the data, will help us be able to monitor the drug supply. So it's kind of taking in a lot of the data, like in the presentation you saw before, where we can detect emerging substances, keep an eye on trends, and then we can take this information, put it into a summary, and then send it out to our community partners so then they can inform the community members. And so overall, our approach is centered in public health and harm reduction first and foremost. So drug checking in practice, this is really about emphasizing and promoting individual autonomy and anonymity. So what we do when we're collecting data is we try and make sure that we're protecting the person as much as we possibly can, which means we don't collect information on names, dates of birth, anything that could be used to identify them. We've also recognized the importance of leading this work through lived experience of drug use and science. So I say these two things together because I think they need to be recognized as being equal parts in this sort of process. Lived experience of drug use is just as important for our work as the science is, because without the lived experience and the expertise of the communities, our science really isn't going to reach the people in the way that we intend for it to. And so also with that in mind, kind of building off of that point, is to make sure we're aligning the science with the community needs. That means that while something might be academic in its nature, we really need to make sure that we're molding and creating to serve the community first and foremost, rather than serving academia or research first. And then lastly, what we're doing is we're kind of taking some of these existing technologies and repurposing them and repackaging them so they're used for public health. So now I'll talk a little bit about the program that we have at UNC, which really stemmed from a lot of this work that the North Carolina Survivors Union started. So once they got this point of care machine, we realized that we wanted to do some more, some confirmatory testing with our lab on UNC's campus. And then from that, we started to also realize that there's this potential opportunity for people to be able to actually mail in samples directly to us. So I'll talk a little bit about that. So this idea kind of started from the work that we had started in Greensboro and that the North Carolina Survivors Union started. But it kind of built off of this other idea of the COVID test kits, noting that we now have the ability to do our own tests at home for COVID. We're kind of using a similar model where we will take a sample of a substance and then someone can mail it off to our lab and we can get their results back to them. So it's a pretty similar type of packaging and all of that type of information. So what it is we have these tiny little vials. They're very small. And we only need a tiny little amount of a substance that is put into this vial with this liquid. It dissolves within the liquid, which makes it so you actually can't retrieve the substance from the vial anymore. So that makes it so this can actually be sent through the mail. And then this, you can take a powder sample. Someone can also swab an empty baggie. Someone can also drop a cotton in there as well for analysis. And so we've kind of started to adapt these kits as we've gone along the way to make sure that they're kind of actually usable in the field. And so this is what they looked like when we first got started. We were just kind of labeling them as we went. But it really showed, this picture shows kind of the wide array of samples that we get after they've been dissolved in this liquid. So it's been pretty interesting to see. And then when someone mails in a sample, they also have the option to fill out this form that collects some information. It's mainly one of the important pieces of information that we get is what the expected drug is. That will help us understand what type of lab tests we need to do. And then it also has some basic information to know if someone had a particularly unexpected reaction to a substance. So that information can be used when we kind of summarize that. It's also been really informative when we had a sample someone turned in who it was supposed to be a fentanyl sample, but the person had hallucinations when they had taken the sample. And we got that information when the sample came in. And so sure enough, we found a PCP type analog within that sample. So that was like one of those instances where when we get that information back out to the person, it confirms their experience. And it also was important for us to keep an eye out at the lab so we can help try and see what sort of explanation there might be. So this is a picture of just a slide with 4 different sample results that we've gotten. These were all, purchased as like heroin or fentanyl related products. But you can see the wide range of results that we get. So sometimes there can be, over 8 different substances that we find within a sample. Other times it can be pretty straightforward where it's just fentanyl, 4-ANPP, which is related to fentanyl. And oftentimes these are cut with sugars as well. And so I wanted to acknowledge the Arrowids. They have been running this website Drugs Data for quite some time. If you're not familiar, it's really interesting to look at the different types of drug samples that are submitted. So they have a mail-in drug checking program where they analyze results from all over the world, mostly in the United States, but they do come in from other places as well. And so these particular results are all ones from Michigan. So you can see there aren't a whole lot of samples that come in from Michigan every year, but they do come in. And so it's just like an interesting piece of information if you want to keep an eye out for other sorts of drugs. There's a very detailed result tab for each sample that comes in. And so that was really like the individual level data. So someone can submit a sample, they can get it run at Arrowid, Drugs Data, and then they'll get their results for their own, to inform their own use. Then there's also other drug checking programs that are developing all over the country. And some of the most established ones have been in Canada. And so they've created these really comprehensive data dashboards that are really working at kind of this community level that I was talking about. So summarizing the data across all of the sites within British Columbia and pulling this together to tell people like, these are the different trends we're seeing. And so it's really informative if you want to keep an eye out for some new substance and maybe you don't have access to drug checking for yourself. This could be the type of information that would be helpful. Toronto also has another pretty comprehensive drug checking service. And they also summarize their data and it's available online. So that's pretty much it. And I am happy to answer any questions you might have.
Thank you, Mary. It was awesome. Yeah, let's, if you got any questions, plug them into the Q&A, and I'm going to go back and there are a couple for Elaine. I just wanted to make sure that Mary was able to get through her presentation. And, first off, it looks like there are some inquiries into your slides being available, Elaine.
Oh yeah, I can share them, absolutely.
Great.
What's the easiest way to do that?
After the summit, we'll put them on the state website. So michigan.gov/SSP.
Okay.
And there'll be a link to the Harm Reduction Summit, and the session's also recorded.
Great.
And then, Elaine, what drug checking equipment does Michigan State Police use?
Great question, especially after the last presentation. So we use 2 of the 3 instruments that were discussed there. We use the FTIR, and then we use the gas chromatograph mass spectrometer. We don't use any of the test strips. I've been really curious about those, but we've never purchased any to try or anything like that. My experience with those kind of tests, though, is that they're of some use, but you can't 100% trust them.
Yeah, Mary, there were a couple questions on that, and I guess Mary or Elaine, what's the utility of the test strips now, that there's so many analogs?
Yeah, I mean, I think they're still a useful tool. They're just not as useful as maybe they once were, like 5 years ago when fentanyl was just starting to emerge. I think that's when we really needed them within communities, and they weren't super widespread. They're not even that widespread right now, but depending on where people are. But I think that there's still utility when you have a sample that you're not expecting fentanyl to be in. There are some issues with false positives for like MDMA and methamphetamine. So that's something that people need to keep an eye out for. And you have to be kind of careful with how much concentrated the sample is within water. But it's still useful for like cocaine-based products and some other ones as well.
Yeah, thank you. And then how much does it cost for an FTIR? Is it difficult to use and operate? Wondering if this could be an effective method if it could be operated for more drug checking at point of service.
Yeah, so we kind of say that you need probably about like $40,000 to buy the machine and to get some of the reference data to back it up on your machine to get it started. So that is a pretty big hurdle, particularly for a lot of community programs and nonprofits. But once you have it, there aren't really big maintenance costs going forward. In terms of running it, we train a lot of people here, and it's kind of like you learn as you go type of thing. You don't need a chemistry background. I've been trained in how to use it, and I don't have a chemistry background. So I think it's mostly like if you're willing to learn and you're interested, then people can definitely pick up the process.
One thing I'll add to that is the FTIR is really good if you have a pure sample. But one thing that it will not pick up is if you do have like fentanyl and cocaine or something like that, that instrument's not necessarily going to tell you that. So the usefulness of it for what you may wish it would do may not be there. Trace amounts of things it will not pick up. It's only going to pick up kind of the primary component of a mixture.
Interesting.
Yeah, we always kind of say that like if something is less than a 5% concentration, that's when you really don't get a reliable estimate of whether something's in there. So that's why we have used like fentanyl test strips in combination, because those ones will kind of pick up that information much more.
And then for either of you, are there more trends for shock dope, which to my understanding is made with RAID, a car battery, and then mixed with meth, for example?
I've never heard of it before.
Yeah, I haven't seen that myself. So yeah, I'm not sure.
Yeah, I guess, Elaine, do you see any samples like that come in where, you see it and you hear what it is and you say, I'm not even testing that?
It happened more when we were in the meth lab world, when there actually used to be meth labs in Michigan. We would see that occasionally. Because if we have something like an acid and we put it on our instrument, it can actually destroy our instrument. So we would have to be pretty careful with total unknown samples, especially if they were in liquid form. But for the most part, if we get even mixtures of powders and that sort of thing, we have different chemical ways to separate the drug from what it's mixed with.
And then, you said that when there used to be a lot of meth labs, when there were still meth labs, I guess, is there, what's the difference between the what you were finding in the testing then and what you're finding now?
So when we were, when I don't want to say when meth labs still existed in Michigan, because there are a few out there, but we've gone from about 2,000 meth labs a year in Michigan to less than 50. So very, very steep decrease in meth labs. And the meth that was produced by, essentially almost always just produced by the consumer, was less pure than the meth we're seeing now. The crystal meth is almost always 100% pure. The samples that they were making would be more in the 50% pure range. And I think there was also the danger of other things being in there that they didn't know about. Meth was almost always made with pseudoephedrine or Sudafed, and a lot of meth still had a lot of that Sudafed left in there as well.
Interesting. 50% to 100%. It's a big difference. And then there's one question I skipped over closer to the beginning. How can community-based organizations and partners engaging in harm reduction work to assist and partners engage in harm reduction work to assist clients with safe drug supply efforts? Are there current policies or programs at the state level to support this work? If not, what could be some best practices and/or programs that could fill that gap?
So we don't really have any part of that, but one thing that I would say is just the sharing of information is so important. The average person in Michigan has no idea that 93% of opioid samples have fentanyl. They still think it's kind of a rare thing. They think it's a kind of a one-off, when in reality, if you buy something you think is heroin, it's almost certainly not heroin. So I think information is absolutely huge. And even people that are buying something that's being sold to them as cocaine. I firmly believe that that's happening and the dealer doesn't even know it, but it's actually fentanyl and not cocaine. So just that knowledge, I think, is so important, that drugs are very, very risky right now, that you really have no idea what you're getting.
Yeah, and I think
your data showed that. And yeah, go ahead, Mary.
Oh, no, I mean, I think with all of this, like, there's a huge opportunity for community-based organizations to be running sort of these like point of care drug checking services. And I think that's probably where they would be kind of best situated, particularly within like community organizations that come from the communities that they serve. And I'm not sure what the kind of support looks like at the Michigan state level. We've been able to get, the North Carolina Survivors Union has been able to get some support through the North Carolina Health Department to support their work. So I think that there's my hope is that there's more opportunities coming through where kind of state health departments will work with harm reduction programs to kind of get more of this information out there, so it's getting to the people that need it the most.
Yeah, I actually met Elaine through a group we recently formed, the Drug Monitoring Initiative, which is a data and information sharing group here in Michigan. And still in its infancy, but we have high hopes on getting two-way information transmission. So we're at 1 o'clock. Again, I sincerely appreciate our speakers, Elaine and Mary. Thank you so much for sharing, and thank you for all those who attended and participated and asked questions. Hopefully you'll be joining us again tomorrow at 9 a.m. And yeah, hope to see you then. Oh, and please fill out the evaluation. I'm supposed to plug that. So thank you again.